I gotta start the recording. Hi, Jonathan. Hope you're doing well. And then I'm gonna do all the tricks that Dr. Annette and Dr. J normally do, which I don't know how they keep up with all of it. Okay. Okay, so I'm assuming you guys online can hear me since you're responding to me, so that is great. So, let's just start. Yeah, I'll just start right here. Okay, so hello, welcome to the study session. You know, we're a smaller group, but that's okay because I kind of prefer smaller groups anyway, because if there's something specific that you guys need help with, we can kind of hone in on that and have more discussions about it. But I guess the first thing that we'll kind of talk about before we get started is like, okay, how should you approach studying for the class and studying for this first exam? So I guess that in part depends on like your learning style. Like in my initial study session, I talked about like learning styles and how those come into play, like how you study. So if you're a read-write person, you're probably going to want to read the textbook and read through the notes and kind of jot down stuff as you go. If you are an auditory learner, I know that that's my style, so I like to get together with a group, and like if there's a concept that I don't understand, I want somebody to talk to me about it, and then once we're finished with that, I want to tell them about it. Because if you can explain it to somebody else, then you really understand it. And then there is visual like graphics and stuff. And then kinesthetic learners, I feel like that's a little tricky. It's mostly athletes, but I feel like just the act of like getting up and like moving around while you're studying, like even if you literally just stand in place and like do like a little dance or something. Okay, here are the different structures of a protein or something like that. So it just depends, but I guess when I was studying for the class, if that's what you're curious about, like how did I do well? Mostly I would go through my notes, and when I took notes in this class, I would directly print off like the PDFs, like the PowerPoints, and I would write directly on the slide. You do that?
I don't know about that.
Okay, yeah. So how do you all take notes for the class?
I print off the slides and write on those, but I also like pretty much just draw on those with what she's drawing, and then take my own notes on my notebook.
Okay, what about you?
Yeah, I just write it down and I add notes and like arrows pointing to stuff, like helpful tips to remember things.
Yeah, for sure. So, yeah, those are definitely good ways. And how do you all think those are working so far? Like, you know, are you finding that it's helpful to do the stuff that you're doing now, or do you think that you're kind of like overwhelmed with like all of the material that you have to write down? And
So it's been a good pace so far.
The exam will tell me if it's a good lyric or not.
Okay. Usually I feel like you can get a good perception of like what you're doing, like if it's working or not. Like usually if you can't keep up in class and you're too busy writing to listen, that's usually a bad sign. But if you can manage like a good flow, you're like listening, but you're able to write and do both at the same time, I feel like that's usually a good indication that it's working. And you can remember it too. Okay. So yeah, that's what I would do. And then for vocabulary, I would use like Quizlet if you like virtual applications, or if you need paper, like actually go to the store and buy some note cards and just kind of dole those out. And I think when I was in this class, I still have my Quizlets from the first exam if you all would be interested in those. It's mostly just vocabulary, but that's mainly what I did. I would just go through my notes. I would attend study sessions, right? Usually the TAs hand out study guides, and I believe that there's a study guide already uploaded to the eLearn page. And I can actually check that right now. So if you go under teaching, like on your eLearn page, and I know that I'm on the section 3 eLearn page, but your eLearn page looks exactly the same. If you go to teaching assistant materials, a bunch of these are just like Amoeba Sister videos, which are super cute, by the way, and they can help you understand stuff. They break it down. But you'll see right here, there's an exam 1 study guide, and when Dr. J finalizes the exam, I'm going to go in, maybe change some things on that, because I get to look at the exam, and I can let you all like know directly, okay, you should definitely know the different types of structures for proteins and stuff like that. Yeah, I will help you target information, and you just have to study, put in the work to get it to memory.
Quick question. Is this that you learned for Dr. J's class, or would this also be available on
That's ours.
Both.
Oh,
Yeah,
okay.
so they are identical pages.
Okay.
But Jasmine and I will work together to update this if it needs updated. So if I pull it up here, there may be some things on here that we haven't talked about yet. So like cell theory, characteristics of cells, you see there's stuff on here that we haven't touched yet. And typically what they do for the exam is that they will lecture up until Friday, and the cutoff for the exam is what we end on Friday. Because that's when your exam will open, and it'll be open throughout the weekend.
Wait, you're saying today she'll come up or next Friday?
Next Friday, because that's
So
when your,
stuff can still be.
yes, unless she specifically says, okay, this is the cutoff for the exam, and usually she'll do that on that Friday.
Wow, okay.
Okay, so just a little FYI, but usually she makes it super clear. These are the boundaries for the exam, and then, you know, Jasmine and I, we work together to make sure those are clear and that you all know exactly what to study in order to do well in the exam. Okay, so we'll update that probably. But yeah, that's pretty much how I study for the exam. So, you know, we will work together to best prepare you. And if you have any questions, like any specifics, if you need to schedule a tutoring appointment, did Dr. Annette show you all how to? Okay, perfect. So yeah, but I guess we'll just go ahead and dive into the material and like what is really important for you to know, because sometimes it's hard to weed out, okay, this is absolutely essential and this is something I can kind of go to the wayside for like a hot minute. Okay, so first thing is the levels of organization, which I don't know about you all, but we spent a lot of time talking about this and like the definitions of them. And what I really want to draw your attention to is starting from the organism all the way up to the biosphere. Those are the really important ones. And remember, according to the Organization of Life, atoms are the absolute smallest and biosphere is the absolute largest. So let's just go through the definitions, or the more important ones. So who can tell me what a population is?
Is that when there's a large number of organisms?
Living together in an area at the same time.
So yes, that's a part of it. So we have organisms in an area at a given time, but I'm going to also add something. It has to be the same species in a given
Communities
area.
is one different, right?
Huh?
Is communities the one where it's different?
Right.
Living together?
Yeah, so a population are members of the same species living in a specific area. So, for example, all of the gray squirrels on UC campus, that's a population. Community is members of different species living in a specific area. So that would be like us, the gray squirrels, the geese, which are very mean, and just all of those different species together. So that's a very important distinction to make, and that will most definitely be on the exam. And that's usually one of the most missed questions because they're easy to mix up, and they all kind of build on each other. Okay, what about an ecosystem? So they're all kind of building on each other with organisms, which make up populations, which make up communities, and then ecosystem.
Multiple communities making up an ecosystem.
Yes, okay, that's a part of an ecosystem, so all of the living things, but what also makes up an ecosystem are non-living things. So like temperature, pH, soil, like all of those different things. So like with ecosystems, you typically hear like tropical forest, tundra, so all of those living and non-living things that make those ecosystems different from one another. And then the biosphere is basically literally everything, the entire world. But that's not as important. Populations, communities, and ecosystems are the more important ones, and you will see those on exams, and you'll also probably see them in lab too.
You said ecosystems, living and non-living things.
Yes.
Oh, that's.
It's just all living and non-living things in the area.
Okay.
So is that clear?
Mm-hmm.
Good. Okay, so another important thing are the characteristics of living organisms, right? Being able to go through this list and evaluate whether something is living or non-living. So there's an extra one that was added on here because the book lists one extra one, the evolution one. But we know that living organisms have one or more cells because a cell is the basic unit of life, right? And we'll actually learn more about the cell in the next PowerPoint we cover in class. They also require energy, and that energy goes towards metabolism and growth. They also respond to stimuli, so something happens in the environment and they're able to respond. They adapt to things in the environment. They also reproduce. And then the evolve is the last one. So just memorizing that list. And this would be an example of a really good like flashcard question. Maybe on one side, what are the 7 characteristics of living organisms? And then you take a moment to try to list all of them and then check yourself. So yeah. And I mean, you could try to come up with like a mnemonic or something if that's your style to remember all of these. But I feel like the biggest thing with this one is that, remember, heartbeat is not on here, right? Because plants are living things, but they do not have a heartbeat. So that may be a trick question presented. Like a multiple choice question, which of the following is not a characteristic of living organisms? They grow, they reproduce, they require energy, or they have a heartbeat. You would click, they have a heartbeat. Is that good? Okay. So at any point, you want to review more or you have questions, feel free to stop me. Okay. So this one, I don't know with your class, but I know that my class had a lot of confusion about evolution just because it's literally such a big theory and it can be confusing when you start talking about it. But essentially all you need to know is that it's basically modified descent of common ancestors. So, over time, the environment changes. And when the environment changes, living things have to adapt to those changes, right? And sometimes those changes make some organisms, a better fit for their environment so they can survive and then reproduce, while it doesn't allow others to live as well. So, of course, those are the ones that die. And that's basically the whole principle of that steampunk or peppered moths example, when the soot from the industrial evolution covered the barks of those trees, so the white moths were exposed. Birds pick those off. And as Dr. Arnett says, you can't have babies if you're dead, right? So the ones that were alive, which were those dark moths, were the ones that were able to reproduce, make more of them, and that's why there was a change in the way that they looked. Did that make sense to you all, like in class and stuff?
That one actually made a lot of sense.
Good. Okay. So, does that make sense to you all, Madison and Jonathan, like the evolution, everything we went over so far? If it doesn't, feel free to unmute yourself or type in the chat. Okay, good. So yeah, I mean, we usually don't get to this unit in class because we have so much to cover in between then, but if we do get into it, we'll talk more about evolution, Darwin's theory, and like how that applies. But basically right now you just need to know like the general principle like this. Okay. Next thing, types of reasoning. So deductive reasoning versus inductive reasoning. And there will definitely be examples, kind of like the activities that you did in class. The Harold is a man, or grandfather, Harold is bald, all grandfathers are bald. Something like that will be on your exam, and you'll have to kind of ascertain whether or not that's inductive reasoning or deductive reasoning. So remember, what I like to do for these is that deductive reasoning is like your detective, right? You have all these little pieces of information, and you put them together to make a single specific conclusion. Like Larry committed the murder. It's a single instance, it's very specific. But with inductive reasoning, which is the reasoning that we see more commonly used in science and scientific methods, is that you take all of these observations from the environment and you make a generalization. So your conclusion is broad rather than specific. So did you all feel okay when you did like examples of inductive and deductive reasoning in class?
Ascertain is what?
Basically, it's gained knowledge.
After we did examples in class, I felt better, but at first it was a little bit.
Yeah, it can be pretty confusing, but I mean, for this, if you want like a good study tip, come up with your own examples and then like compare them to what you did in class, like, or you can search up examples like deductive reasoning scenarios or
What you
inductive.
said was that was inductive, wasn't it? The grandfather one?
Yeah, because that one's
Yeah,
really
it's
broad.
yeah.
Yeah, like not all grandfathers are bald, right?
I know it then, okay.
Yeah. Good. Yeah, so you just need to basically know the definition and know how to apply it. And that's usually, I think there's like a question apiece on this. Okay. This is something I believe we weren't able to get to in lecture, but they were able to get to it in lab. But it's very important to know the definitions of these. So a hypothesis, you always hear as an educated guess, but we're going to take that one step further and make it sound a little bit better. An educated prediction based on observations and or previous tests. And the two requirements for a hypothesis is that it must be testable and falsifiable. So testable is obviously, you know, can I do maybe like an experiment to test, you know, what's going to happen. And then falsifiable just means that it can potentially be proven false. So usually with this, I think Dr. Annette says something like, can you prove that there is a flying spaghetti monster in space? Yeah, like you can't do that, right? So if I was going to say, you know, make some hypothesis revolving around that, that wouldn't be a very good scientific hypothesis. But if, for example, I wanted to test Alka-Seltzer in different kinds of water, right, and whether that affected the time of dissolution, maybe that would be a good experiment and hypothesis to do. Okay, and then other definitions are law and theory, and definitely the difference between law and theory. So a law basically is a generalized statements based on repeated observations and tests, and they can be used to predict future events because they basically occur like clockwork, like they always happen. Like, do you ever hear anybody question Newton's law of gravity? Especially if you're on Earth, right? Because guess what? If you hold something up and you let go of it, what does it do? It falls to the ground because of gravity. So when I think of law, I think of something that always happens and you can use it to predict. And then a theory is a little different because it attempts to broadly explain, and it actually collects groups of laws and observations together. So when we're looking at the scale of like how narrow or broad these things are, hypothesis is your smallest, right? And then you law, and then your theory, which groups all of those into one umbrella. So when you think of theory, I mean, you may think of different things. We'll eventually touch on cell theory, which is a type of theory. And there's the theory of evolution, right? You think about how broad that is and how much goes into that. Are we good with this? Okay. So just knowing the definitions, essentially. Okay. And I know that this was also a source of confusion for a lot of people in my class as well, like the control groups and how to tell if something's an independent variable or a dependent variable, which can be challenging at times, depending. But I know that some of the e-learn homeworks and like your labs give you experience in, assessing these. So basically the purpose of a controlled experiment, so like why do we have controls, is to minimize the effects that are not being tested. So it basically increases the reliability of the study. So if we're able to control everything, we can say that this variable leads to this variable without outside influences. And that's really important in the experiment because we can show cause and effect, right? So some definitions. There's a difference between a controlled environment and a control group. So a controlled environment keeps all baseline information the same, and a control group is basically a group that doesn't receive the treatment. So for example, let's say I'm growing corn, and over here I have a plot of corn, and then over here I have a plot of corn. Let's say that I keep everything the same, right? I'm going to give them the same amount of water, the same amount of sunlight, the same amount of soil. That's so I have a controlled environment. And then let's say over here, for one group of corn, I'm going to give them fertilizer. And then the other group, I'm not going to give fertilizer. That would be my control group, the one that doesn't receive the fertilizer. Because in this experiment, I'm trying to see the effects of fertilizer on corn growth. Okay. So in that same experiment, if I'm assessing the effects of fertilizer on the height of corn, what would my independent variable be?
So on the height of corn?
Yeah, if I'm trying to show whether fertilizer does or does not improve the growth of corn.
So the fertilizer would be independent?
Right, so the presence of fertilizers. I would have two groups. One group doesn't have it, one group does. And so my dependent variable would be what I'm measuring, right? So like
the farm knows.
right, like the plant height, for example. So independent variable is usually the variable that is altered and that directly causes change in your dependent variable, which is usually what is measured as a result. So for this on the exam, typically like the definition of a control group and then like definitions of independent, dependent variables, and you may be presented with a scenario like I just presented you and ask, what's the independent or what's the dependent variable? So is all of that good? Because that basically covers that like full introductory PowerPoint that we did, and we're about to move into biomolecules. Okay. So biomolecules, this was a really fun PowerPoint. There was a lot of really good information, some of it which may be frustrating, but we're going to try to break it down a little bit. So we have 4 major types of biomolecules, right? We have our carbohydrates, our lipids, proteins, and nucleic acids. I know that you all haven't got to nucleic acids yet, but I'm going to go ahead and give you a preview into that. So hopefully some exposure now, and then you'll go to class, will jog your memory, and hopefully help you remember things better. So first thing, with carbs. So the in-class assignment that you're working on right now, where you list like the functions and uses of carbs in the body, and then like unique characteristics, that's a really great study guide for the exam. So definitely recommend, of course, doing that and turning it in because it's 4th grade, but also because it's a great study guide.
I'm sorry, you said to do what? Which document to do?
Oh, just like I'm talking about your in-class activity that you're doing right now.
Okay.
It's worth a grade, so you should definitely do it and turn it in, but it's a great study guide
Okay.
for this part of the exam. So carbs are used as fuel and energy for the cell and also building materials, so you should definitely know the uses. And then also, when we talked about carbs and basically introduced biomolecules, we talked about the differences between monomers and polymers, right? So what's the difference between those two?
Monomer is one ring, and polymer is a chain of rings. Or it can be many.
Yeah. So yeah, a monomer is simply, like you said, a single unit, and a polymer is many units. So for our carbohydrates, then the monomer would be a monosaccharide because the suffix saccharide just means sugar. So that word itself means one sugar. And we know that our identifying characteristics of carbohydrates are that they have lots of hydroxyl groups. So you can see in the long chain structure and the ring structure, you see all of those hydroxyl groups. And we know that the presence of a lot of oxygen is going to make things polar or hydrophilic, right? That's really important to remember. And another functional group that we have is our carbonyl group, right? This right here. And the functional groups are really important because she may give you, for example, let's say she puts this on the exam. What kind of biomolecule is this? And you would have to select from carbs, lipids, proteins, and nucleic acids. And by looking at it, you're like, oh, okay, this is a ring structure. It has a lot of hydroxyl groups. This is probably a carbohydrate. Okay. So another thing that we talked about with like the monomers and polymers, so if we have two sugars, it's a disaccharide. And then when we continue adding, so 3 or more, polysaccharide. And how we ultimately link those two together, and this is true for all of the biomolecules, so like lipids and our proteins, that we do that through dehydration synthesis, and if we want to break them apart, we do that through hydrolysis. And this is actually going to be really important because you're expected to know these reactions too. So dehydration synthesis, if we break that word apart, dehydration means what?
Take away water.
Take away water, right? And synthesis means to create. So by removing water, we can create something. So dehydration synthesis just means that we're taking two smaller units, in this case, 2 monosaccharides, and by removing water, so water is H2O, so we can take a hydroxyl group from one sugar and a hydrogen from the other. That's water, pull it out, and we form a covalent bond between the two, and that's dehydration synthesis. Now, let's say that we start out with our disaccharide and we want to break it up into 2 monosaccharides. That's hydrolysis, right? Because we know the prefix hydro means water, right? And then lysis means to break apart, right? So through the addition of water, then, we add water, we can restore the hydroxyl groups and then have our 2 monosaccharides. So is dehydration synthesis and hydrolysis clear? Because that's a really important part.
For the diagrams, what would do, like, would you suggest making like a puzzle set to learn all the, I don't want to say shapes, but like the diagrams and hexagons and stuff that go with these?
I mean, yeah, you can definitely do that if you want, but for the hydrolysis and the dehydration synthesis particularly, and I think what they typically like to do, they like to do it with carbs, and they also like to do it with proteins. Lipids are a little bit more difficult to do with.
Yeah.
But just kind of memorizing that we always have these hydroxyl groups that are being combined. So in the end, with like hydrolysis, we need to restore them. And then if we're doing dehydration synthesis, water needs to come out, so we need to link.
Okay.
Does that make sense? But yeah, as for like the different shapes, because you know that's what they look like at this point. But if you take like other chemistry courses, like organic chemistry, you learn more about the ring structures and stuff. But it won't be as heavy on your exam. Like she doesn't expect you to count like all the carbons, hydrogens, and oxygens. Like that won't be a thing either. Promise. Because I didn't learn how to do that until my sophomore year in organic chemistry. Okay. So this brings me to my next point. If you are struggling with dehydration synthesis and hydrolysis, there is a great worksheet on both eLearn pages. I think it's under week 2 and week 3. And you can click on it and just kind of go through this, but it's basically just that same process that we just did, like define hydrolysis, dehydration synthesis, identifying that Janet is a carbohydrate, and then just kind of linking them up. So I would definitely recommend looking at that. It's great practice. And like if you have any questions about it, like we won't per se go over it in class, but Dr. Annette, Dr. J, me or Jasmine can answer any questions, or if you just want your work checked. Okay, let's go into lipids. So lipids are our next type of biomolecule, and these are typically used for energy storage and structure for the cell, like we learned about phospholipids, which are integral to the cell membrane. And we learned that there are 3 types of lipids. So we have triglycerides, which consist of plant and animal fats. So, you know, personally, my parents go to the doctor and they get their triglycerides checked, right? That's a common thing. But other than those plant and animal fats, we have phospholipids, like I mentioned, that are important to the cell membrane. And then we also have steroids. So what's in common with all these lipids, right, is that most of the time we have these long hydrocarbon chains, right? And those hydrocarbon chains, we learn carbon and hydrogen make things nonpolar or hydrophobic typically. So triglycerides specifically, which I don't think you have to know this as much, but they're composed of glycerol, which is this backbone right here, and then 3 fatty acids. And then you have ester bonds, which that's a new type of bond. Like with our carbohydrate, we had a covalent bond. Here we have an ester. And then, like I mentioned, those long hydrocarbon chains are ultimately what make the triglycerides nonpolar and hydrophobic, which means that they're water-fearing, right? And that is in direct contrast to our hydrophilic carbohydrates, which love water because they're polar. So phospholipids, this is the important one. If you don't remember anything else about lipids, remember the phospholipids because they will come back. So we know that phospholipids also have glycerol and they have fatty acids, but what really makes them important is that they have that phosphate group, right? And because they have these hydrophobic and hydrophilic regions, that's what ultimately allows them to form that lipid bilayer that makes up the cell membrane. So this just shows you, doesn't necessarily show you like where the phosphate and stuff is, but you can kind of guess. Is your phosphate, which has a bunch of oxygens, it's going to make it polar. It's going to be hydrophilic. So your phosphate group is up here, and then your hydrophobic tails are right here. So cells themselves are mostly water, and then on the outside of the cell, there's water too, right? So our hydrophobic tails are going to orient themselves like inward because they don't want to be near water, right? And then our hydrophilic heads are where the phosphate is. they want to be near water, right? Because like dissolves like.
Can I pay attention to the last slide?
Yeah, and I'm going to send this PowerPoint out to everybody, so you'll have a copy of it. But if you still want to take a picture, you can. Okay, you're welcome. So everybody clear on like triglycerides and phospholipids? Okay. So the final one is a steroid, which if you take any anatomy classes, this is when steroids and hormones become important. But we just wanted to briefly introduce them. So they're basically signaling molecules. And the big thing that you need to know about steroids is that they always have these 4 fused rings. And they may have different groups sticking off of those, but you won't need to know about those. Just, for example, if you get a picture on the exam that looks something like this, and let's say it has a bunch of groups sticking off of it, which those won't matter, and which type of biomolecule is this? Is this a carbohydrate, a lipid, a protein, or nucleic acid? Then you would definitely click lipid, right? Because you would recognize it as a steroid. Okay. So, good on lipids? Okay. So then proteins. Proteins are probably the most important, right? These are our favorite biomolecules, as everybody likes to say. And if there is a lot of questions on the exam about biomolecules, you can bet it'll probably be about this one. So we know that proteins are the most versatile, right? They do a lot of things. They can act as enzymes, storage, structure, protection, transport, and regulation. And I would just, memorize a few of those functions, go back to your in-class assignment. And we know that proteins, which are also called polypeptides, are basically made up of amino acids, right? And we know that there are about 20 amino acids. You will not have to memorize the structure of all those amino acids. I promise. You won't even have to memorize them in biochemistry if you take that. You just need to know that amino acids make up proteins, and that all of the amino acids are different because they have different side groups for that R group. So, unique things about the structure of an amino acid. We have an amino group, right? We haven't seen that before. We have a carboxyl group, which we've seen that before in lipids, right? And then hydrogen, that's not new, that's in everything. But this R group, it can be a bunch of different things, and that's basically what generates the diversity. That's why you have 20 different ones, because they all have different R groups. And it can make it nonpolar, it can make it positively charged, negatively charged, but you won't have to know about any of that. And then another important thing with proteins is that structure ultimately determines function. And if you mess up the structure, then you completely mess up the function. Okay. So then you might be asking, how do we link these together or take them apart? And we do that through dehydration synthesis and hydrolysis, right? And here I've just shown the process of dehydration synthesis. And then if you wanted the hydrolysis, right, you go in the reverse direction. So same sort of concept. So we have 2 amino acids here, right? And we want to link them together, form a peptide bond through our dehydration synthesis. So we need to take out water, right? So it's always going to be the hydroxyl group from the carboxyl, and then a hydrogen from the amino. And then we form the peptide bond. So it always happens like that, I promise you.
The peptide bond is the one with the nitrogen on it?
Yeah, so it's basically where it's formed. So you always have like this carbonyl, the C double O, and then a nitrogen right there. That's your peptide bond between that carbon and that nitrogen. And then of course you have your water over here because it's being released. Okay, is all of that clear? So then, is this where you all stopped in lecture? Like the level? Okay. So I'll kind of introduce the quaternary, and then you'll get some exposure to that in lecture, and then we just got to talk about nucleic acids really quick, and then I will let you go and you can enjoy your weekend. But, okay, so with proteins, you know, we have different levels of organization, different structures. We learned that the primary structure is basically that linear amino acid sequence that's just simply held together by those peptide bonds, right? So I think Dr. Arnett does like a little paper folding exercise, right?
Not really.
Dr. J used like a phone cord, you know, one of those ones that like curls up. And if you stretch it out, then it's basically your linear sequence. And so for each of these, you basically just need to know like what's the definition, what does it look like, and then what is it held together by. So for primary, it's a linear sequence, and it's held together by peptide bonds. So for secondary then, we could either have our alpha helices or our beta pleated sheets. And these are held together by hydrogen bonds. Our tertiary, which Dr. Annette usually says is represented by the headphones that are nice and neat that we put in our pocket and then only to grab them later, and they're a tangled mess. So basically you have everything folding in on each other because of those side chain interactions. And at this point, the protein is three-dimensional, which means it's functional. And this is the stage that most proteins reach. Some proteins have a quaternary structure where they basically they need multiple proteins. So I know a good example, and it's in your notes, is hemoglobin. So you have like 4 separate proteins that come together to make one, and those are held together by ionic or hydrogen bonds. Like I said, for these, know like the definition, what does it look like, and then know what it's held together by. So basically this slide is all you need to know about the structure of the protein. Does all of that make sense? Is there anything that's confusing to you or anything like that? Okay, so let's finish this up. Let's talk about nucleic acids briefly. So there are 2 types of nucleic acids. There is RNA, which is ribonucleic acid, and it is single-stranded and primarily used for making proteins in the cell. And then we have DNA, which everybody's heard about DNA. DNA is deoxyribonucleic acid, and it's basically used for long-term storage of genetic information, and it is double-stranded. So here we see kind of the difference in the structure. And the common thing about nucleic acids is that they're made up of things called nucleotides, which you'll talk more about these in class, but essentially a nucleotide is made up of a 5-carbon sugar, such as deoxyribose if it's DNA, or ribose if it's RNA, a phosphate group, right? We know what a phosphate group looks like because we saw that in our lipids, and a nitrogenous base. And the nitrogenous bases are ultimately what make RNA and DNA different from one another, besides like the single-stranded and double-stranded. So for DNA, the nitrogenous bases are adenine, thymine, guanine, and cytosine, so ATGC. And then for RNA, instead of thymine or T, we have uracil in our RNA. But like I said, you'll talk more about that in lecture. And if you have any questions or there's any confusion, please feel free to email me or Jasmine. And then we'll have study sessions next week to kind of go over exactly what's on the exam and answer any questions that you have. Are there any questions from online? Are you all doing okay? Is everything clear to you? Okay, any questions here?
Could you show one slide?
Yeah, sure. And I'll send this out to everybody who attended, and there's a recording if you want to go back and watch it or anything like that. But yeah, I mean, that's all that I had. Hopefully that was helpful to you, cleared up some confusion. Do you have any questions for me? I mean, just about the class in general or the university in general? I mean, I've been here for a long time.
Well, each chapter has like about this amount of content each time, or is it gonna load on?
So I think it depends. I know that some, like the cell PowerPoint, I know that that one has quite a bit in it because there's a lot of organelles that we have to cover. But I don't feel like they usually cut some stuff if they feel like it's too long, but I don't think any of it, they're gonna like load it up and overwhelm you all. They definitely try to consider that. And we also weigh into, we're like, no, please don't.
Right.
Yeah, we definitely advocate for you all, so. Okay, I'm gonna stop it.